All right, it's officially 12 o'clock, so we're going to get going. Welcome to our second session of Getting into the Weeds 2025. Today we're going to be looking at some research happening in insect management. We have a fantastic lineup with Deb Moreau, Caitlin Congdon, and Janelle McKeel. So yeah, really excited to get going. There will be pesticide points and CCA, CEU points available today, so we're just going to talk a bit about that, and then we'll get going on the content. So I'm not going to spend too much time going over our Zoom housekeeping rules. I am guessing that we're all pros by this point. But I will point out that if you are looking to get pesticide points today, just make sure that the name that you have on your Zoom account is the name that matches your pesticide license name. If you go to your little box, your little square, there should be a little ... next to the mute button, and that should give you the option to rename, just in case you need to fiddle around with that. So for those who are looking for those pesticide points, every participant needs to watch from their own device. So we can't have multiple people trying to claim points over one computer. And I will be posting a link in the chat at the end of the session. You're going to need to fill out that survey, basically your name, your pesticide number, and then print that screen as your sort of proof or your receipt that you attended the session. If you have any issues with that, please feel free to email myself or your favorite Perennia specialist, and we will point you in the right direction on how to get that sorted. We'll also have the QR code for our continuing education units at the end of the session. So with that, we have 3 speakers today, and it looks like we're going to be talking about 4 different insects. I think we will plan to do a question period after each presentation, and then we can also circle back at the end if something comes to mind, or we kind of just want to go a little bit deeper in on a discussion. So I made the executive decision today that we are going to go in alphabetical order. So Caitlin Congdon is going to get us started talking about European corn borer.
Great, thanks, Talia. It's shared up here. All right, great. Well, thank you very much. As Talia said, I am Caitlin Congdon. I'm the field crop specialist at Perennia, and I am going to talk a little bit about European corn borer today, especially in a horticultural context rather than just with corn. So despite the name, European corn borer, it is a pest of a lot more than just corn. European corn borer has a multitude of host crops and has been known to cause economic damage on potatoes, tomatoes, peppers, newly planted apples, hops, hemp, and more. It's also known to be a pest, though not an economical pest, on a number of other crops as well, including small grains, so cereals, and a lot of our common cover crops, including like Sudan grass or sorghum Sudan grass, and even clover. So European corn borer really doesn't discriminate. It'll eat anything, and therefore it can be a problem for everyone. Because what we normally think about corn borer as a corn pest, that is what's most common when we think about what the damage would look like. But I did manage to find some horticulture photos of the damage. So just starting over on the far side there, we've got damage on newly planted apple. And then moving over, we have damage on potatoes and tomatoes and peppers. So the primary feeding damage of European corn borer is really the issue, because it bores into the stem or the stalk or even into the fruit of those different crops, and disrupts the movement of water and nutrients through the plant. The point where the larvae bore into the stem or stalk is weakened, and that can lead to breakage. The common one that we see in corn is it'll lead to lodging because it bores in at that point, and then the stalk is weakened and it tends to bend over where that entry hole happened. So you would see a similar thing in some of these other crops as well. The other thing would be death of the plant above that point. So if you think of something like hops, if it bores into the stem at a certain point and blocks off the flow of nutrients and water, then anything above that is kind of going to go kind of going to go downhill pretty quick. So feeding damage as well as just causing issues with the structure of the plant and potentially impacting the yield, can also provide an entry point for pathogens. So it can increase disease issues, which may further stress the plant or impact yield in its own way as well. So why is this interesting? Why do we care about this? If you grow any one of those crops, maybe you've seen a few corn borer over the years, but it really hasn't been super predominant in a lot of crops over the last 25 years or so, and there's a really great reason for that. So BT technology was introduced in corn in the mid-1990s. So BT is just short for Bacillus thuringiensis, which is a soil-borne bacteria that produces insecticidal proteins. So they developed this transgenic corn that was able to produce these proteins and therefore kill the European corn borer. It's been super, super successful in managing European corn borer in corn crops. And so over time, we've seen a significant reduction in ECB populations across North America. And that can be attributed to the widespread use of BT corn technology. So you can see that would have a huge benefit to other host crops as well, just overall population dynamics really going down. However, we are starting to see a change in that. And in 2018, the first BT-resistant European corn borer populations were discovered in Nova Scotia, and that was the first documented field-evolved resistance that was ever. So across North America, of all the places that they grow corn, it happened in little old Nova Scotia. And since that time, we have seen resistance cases in New Brunswick, one location in Quebec, one location in Manitoba, and now in Connecticut as well. So it's moved across the border. There are multiple BT proteins used in corn. So if you think about them kind of like an active ingredient, as you would in a herbicide or insecticide, there's 4 of them. So they wouldn't all be used at the same time, but they would be pyramided. So that would be multiple proteins targeting the same pest, or stacked. So multiple proteins that target different pests. We are seeing resistance and reduced efficacy with all 4 of the ones that target European corn borer. That doesn't mean that they're not working at all, but it does mean that we need to really take steps to try to slow down any more resistance development. So IPM is integrated pest management is going to be super key. If we don't have other management practices in place and BT control continues to slip, those European corn borer populations are going to rise and become a more significant issue in other crops as well as corn. So what do you need to look for? The first and key thing in any IPM system is scouting. So you're going to scout for egg masses around peak flight timing. There are 2 types of European corn borer. So a univoltine, meaning they have 1 generation per year, and a bivoltine, meaning they have 2 generations per year. From the monitoring that we've done here in Nova Scotia, it looks like we probably have both types. And the biggest difference between them is how quickly they develop. So we would expect to see the first generation of the bivoltine type reach peak flight earlier in the season, so around 300 to 350 growing degree days, which is usually around early July for us. And then the univoltine would reach peak flight around 650 to 700 growing degree days, which is usually late July. In corn, we expect to see larval feeding, so boreholes, tunnels, and things like that in mid to late August. But back to our scouting, you're going to be scouting for egg masses at those kind of peak flight timings, because that's when there's the most moths out flying around and laying eggs. They tend to lay their egg masses on the underside of leaves, and in corn near the midrib of the leaf. And you can see the picture here on the side, and they just look like little clusters of overlapping fish scales. They would have 10 to 30 eggs per cluster. As they get closer to hatching, you'll actually see the black head capsules in the eggs. And then once the larvae hatch and they start to feed and tunnel, then the signs that you're going to look for are frass at the entry holes, and it kind of looks like sawdust. And then as that goes further, you may see broken stems or stalks, and the larvae themselves, which are a whitish cream color with black heads. So we are monitoring for European corn borer moths using pheromone traps at around 10 locations across the province each year. This information is tracked on the Great Lakes and Maritimes Pest Monitoring Network, which focuses on European corn borer as well as other major corn pests. However, some of the traps that are being tracked on this network are in other crops as well, so potatoes and things like that. Since ECB has so many hosts, this part of the site is actually relevant to a lot more than just corn growers, and can be used to track moth capture in real time. Unfortunately, it's not a perfect system, and it relies on the moths being attracted to the pheromone traps. And one of the difficulties that we've come across is that the pheromone lures that are available just don't seem to be working as well as they should. We are also participating in a trap type and lure type comparison to figure out the most effective combination. The typical ones that we've been using so far has been a Heliothus style trap, which I have a picture of on the next slide, and the commercially available E or Z type lures, which are changed out every 2 weeks. If you happen to be interested in trapping for European corn borer on your own and want to contribute that information to this network, please feel free to contact me and I can point you in the right direction for trap and lure suppliers, and help you get set up on the Great Lakes and Maritimes Pest Monitoring site. So just a quick overview of some IPM for European corn borer. We've already talked about scouting, but of course that's our basis for all of this. Insecticides are an option. However, BT is actually the active ingredient in a couple of products like BioProtect and Dipel. And some of the, I believe BioProtect is certified organic as well. So that if we see this resistance continue, then that could be off the table as an option. The other thing with insecticides is timing is super key, because once the larvae bore into the plant, then they're not going to be touched by any insecticide. One that was really widely used before the advent of BT corn technology was stalk destruction. Based on previous research, that's really the best option for corn. The last time there was research done on that was in the mid-90s, and the equipment that we have available has really changed a lot since then. So there is um, current research being done looking at more modern technology like high-speed disks and things like that. Over in PEI, they've actually developed a potato vine crusher for European corn borer management, and that it just goes on the back of the potato harvester. The vines go through it and crush any corn borer so that they can't overwinter. The other interesting option is Trichogramma, which is a parasitic wasp, and it parasitizes the egg masses of European corn borer. It's used pretty extensively for sweet corn production in Quebec, and it seems to be working quite well for them up there. Timing is going to be super key on that as well. So monitoring growing degree days and really scouting to make sure you get there when the egg masses are there. And unfortunately, this isn't a great option for field corn production, just because it's expensive and labor intensive. But it could be a really great value or a really great option for high value crops. And that's all I've got for right now. But if you have any questions, I am happy to answer them, or feel free to reach out to me afterwards.
Awesome. Thank you so much, Caitlin. That was great. I have so many questions for you. Okay, so you kind of alluded to at the beginning, that maybe there's still an opportunity to maintain BT corn as an effective strategy if we're able to sort of manage the development of that resistance. Is that right?
So I would say the cat's out of the bag on it's we're never going to be able to walk it back. Like, we know we
Mhm.
have resistance now. All we can do now is try to slow it down and maintain the efficacy that we have left.
Okay.
And the more that we can do that, the better for us. Also, if we can try to reduce the spread. Obviously, it's kind of sucks that it's our problem here in the Maritimes, but we also don't want to spread that to other people. So the more we can do to just slow it down and try to maintain those traits, maybe it'll give them more time to develop something new for us.
Yeah. But it's a big and pretty immediate shift to a different approach to managing European corn borer.
Absolutely. And because it's
Yeah,
worked for so long, it's really it's a complete mindset shift. There's a lot of us have never really had to even think about the full scale of European corn borer damage in corn, never mind any other crop. So
absolutely. What was the name of the parasitic wasp?
it's trichogramma, and I can I'll put a link
Post
with
it in the
more
chat.
information about that. Yes.
That'd
Yeah.
be awesome. Thank
Yeah, for
you.
sure.
Um, so you talked a little bit about peak flying times. Where
Mhm.
would be the best place to get information on that?
So we have a European corn borer like general fact sheet, which I can share the link to as well, and it has the rough growing degree day timings.
Mhm.
And if you are familiar with farm data tools on the Perennia website, then that has a growing degree day calculator on it. So you can use that to calculate growing degree days for your area and hopefully line up with what we would expect to see for those moth flights.
And so once we have a rough idea of what our growing degree day is and when we think they're going to be actively flying, um, we should be getting out and scouting. Like, is that sort of the best?
Yeah,
Okay.
definitely. Yeah.
Yeah. And are they, I know for some pests, sometimes they are preferential to like new tissue, old tissue, like top of the plant, bottom of the plant. Do we know anything about that for ECB?
Yeah, so they don't really discriminate around crops. They
Okay,
don't
they're
really discriminate around where they get access.
yeah.
Um, yeah, but in corn, actually, we used to find them much higher up, and in recent years, we're finding them all the way down into the brace roots of the corn plant. So they could
Wow.
really be anywhere, unfortunately.
Okay. So like it's probably worth doing the full plant scan in that case.
Yes. Yeah.
Very interesting.
Yeah.
Um, and then I was curious about, you talk about the potato vine crusher in PEI.
Mm.
Was that something that was developed or like is in the process of being developed in response to European corn borer, or was that
So
sort of a
yeah, so it wasn't developed in response to the resistance issue. It was just they have European corn borer in potatoes over in PEI, and of course it's such a huge industry for them. So it was something that was developed through AAFC over there. I honestly don't know that there's something that's commercially available, but I think that the idea is available and didn't seem to be too complicated to implement.
cool? So yeah, I think that is kind of it, unless anyone else, you
Perfect.
can continue to post either in the chat or the Q&A. Um, oh, okay. We had something just pop in the Q&A saying
Okay.
Trigogramma are egg parasitoids.
Yeah.
So it might be useful for folks to take a few samples of eggs when scouting, stick them in a vial, and see if there is any parasitism occurring naturally.
Hmm.
It's a cool little test.
Yeah,
I like
absolutely.
it.
Yeah.
Awesome. Yeah, so that was great. I think we will switch gears then to Deb. But yes, if anyone else has questions for Caitlin, feel free to keep them coming in. And I think there's going to be some goodies in the chat there for you. So stay
Thanks.
tuned. You'll just want to switch the view there, Deb. I did the thing again.
All good.
Beauty. Good to go. Yes.
Perfect. Thanks for the invite. So I'm going to talk mostly about spotted wing Drosophila, and then switch gears a little bit at the very end. But and I'm sure this is this is a well-known pest for most people on this call, but just a little background that this is an invasive species, and certainly we had the first record for detection in Nova Scotia in 2011. Regular insecticide applications at this point in time remain the most effective tool to manage this pest. And certainly feedback from colleagues and my own observations and grower reports suggest that pest pressure just continues to increase. So of course, with that, that is what's driving the spray schedules, especially in berries or any of the sort of tender fruits. So a little background. The problem with spotted wing is that just to kind of take two steps back, it's a small fly. It's a vinegar fly. So it would be, often we call them fruit flies. They're kind of related to the fruit flies you would see, if you discard a banana peel on your kitchen counter or enjoying a nice glass of wine in the summer months. You might see these little guys kind of hovering. So it's a relative of those. They're not very long. The adults are about 2 to 3 millimeters long. They're kind of like yellow, brown yellow, large red eyes. They have what makes this distinct compared to the others is that the females, as you can see here in the corner, they have their egg laying apparatus, or what we would call ovipositors, actually has a serrated edge. So it's a very sharp saw-like edge that allows them to cut through the skin of berries. And then once they have access to the inside of the fruit, then they insert that ovipositor and they lay their eggs directly in the fruit. So you can see this fantastic image here with multiple eggs having been laid. It's actually from Erica Bent. And you can see, if you look very close, you can see small white filaments sticking out of the holes, which are just the little respiratory filaments that are attached to the egg. But you know, once the eggs are laid, the fruit becomes basically unmarketable, because as the eggs hatch and the maggots or the larvae develop inside the fruit, of course, then they cause the fruit to sort of soften and collapse. There can be other problems associated with, you know, juice leaking out, things like that. So the fly can ramp up pretty quick. It can go from egg to, you know, through 3 larva stages or instars to pupa to adult. You know, at 18 to 20 degrees, it can cycle through that in a couple weeks. And in individual females are thought to, you know, be able to lay, you know, anywhere between 300 and 400 eggs. So this becomes a huge problem. I don't want to say chatter, but lots of observations on farms that the pressure is increasing. So I'm not going to go into this heavily, but this graph was produced by Jeff Franklin at AEFC, and it's based on January minimum temperatures from the Kentville, the Environment Canada weather station that's on site here at Kentville Research Center. So these are January minimum temperatures by year going back as far as 1914, and then takes it right up to this year. And why this is important is if you look at there's a sort of a trend line here. So 2 things I think are very interesting with this trend line. If you look at this first kind of this end and compare it to this end, what you can see is an increase in sort of the average minimum January temperatures over the 100+ years. So, you know, we've heard lots of people speculate there's about a 2-degree difference, and we're certainly seeing it in these trends. For me, what's fascinating is if you look at this, you know, sort of just before the late 90s to today, the rate of increase. So the slope of this trend line is actually suggests that the rate of warming is actually increasing. There's a broken line or perforated line through here. This is Kimura 2004. So Kimura was a researcher who published in 2004, and basically he reported, or the research reported, that spotted wing could actually survive average January temperatures of -5 degrees. So what I find fascinating about this is that if you kind of watch my cursor, right about here is when we detected them in Nova Scotia. And if you were to kind of draw a line up here, you would see that the vast majority of data points since that time have been above that perforated line. So it certainly makes a compelling argument that that our winter temperatures, certainly our minimum temperatures, are definitely support overwintering success of spotted wing. The other thing we're seeing is if we look at, again, this graph, the credit goes to Jeff Franklin, who produced this is growing degree days. And again, it goes back to 2014 up to this year, or to the data that we have. So this is, we use calculation of growing degree days. For insects, we typically use base 10 calculation. But again, what you can see is that over the 100+ years, we are seeing an increase in sort of heat unit accumulation each year. In 2011, when we first detected the pest, it was a bit cooler, but by 2012, we saw a very warm, hot spring. And we saw that subsequent field season, we saw, you know, a 40-fold increase in spotted wing picked up in traps that we were sort of monitoring that year. So what does this actually mean? In 2011 to probably 2015, I would have estimated that we were probably seeing 3 full generations of spotted wing in Nova Scotia. It wasn't to say that we weren't seeing, you know, some at the end of a growing season, we were starting to see maybe the development, or some started moving into the 4th generation. So eggs had been laid and they were hatching, but they often weren't getting through the full generation. In 2012, we exceeded 1,400 growing degree days. And that year we had, huge pressure, heavy pressure. And we're continually now seeing that kind of, that the increased trend in heat units, which may be good for other things, but certainly, especially for invasive species, this kind of spells trouble in that it may mean that that we're seeing extra generations, and especially coming out at a time where fruit, certainly like high bush blueberry, where we have late cultivars, could be more disproportionately impacted. So I thought I would focus on sort of how we manage the pest to date. Of course, we're, right now growers are reliant on sprays. But we do a lot with monitoring, and monitoring is done largely through trapping. There are some challenges, and they're faced with trapping spotted wing, and there has been a lot of work on traps, sort of the development of trap, revision of traps in terms of best colors, contrast, where to put them, lures, all of those things. I'm going to focus on 2 trap types, but there are tons out there. If you were just to Google commercial SWD traps, you'd find all kinds. So in the center, the red solo cup with the sort of black tape, which has little 1/4-inch holes kind of punched in around it. The black is really just to enhance contrast, so the flies pick up on that. And we have historically, and I know a lot of the growers use apple cider vinegar, that we kind of bait those traps, and the flies pick up that odor and then go into those holes, and in theory, drown in the apple cider vinegar. Our research has explored other things. We've enhanced the apple cider vinegar with yeast, ethanol, different things to try to sort of improve efficacy over time. But at the end of the day, this has been our tried and true trap. Lots of research has suggested that it's that even though it's effective at catching spotted wing, it's probably a very conservative estimate of what's actually out there. So newer traps have been coming out probably since 2015, 2016, where they're using lures. You can see it here. This is a red sticky trap, and then the associated lure up here. This is a trace lure. So Erica Bent's company did some work this year where they sort of compared. So you can see this kind of apple cider vinegar setup just over here in the corner, where they sort of compared these 2 traps. So you would basically put these traps out, and then you would look at the sticky trap. You'd bring the sticky trap in every week, and you'd kind of look at that sticky trap to see if the spotted wings were stuck to it. With the apple cider vinegar, you would basically dump those traps on a weekly basis, and you would look for spotted wing. So the challenge is that as researchers, we kind of look at how it's easy for me to go and collect these traps and bring them back in and sit under my microscope at my leisure, and figure out what's in the trap, how many females, how many males. But that is not a luxury that most growers have. They're looking for sort of more timely information so that they can kind of plan the spray accordingly. So what we have typically advised is that we're looking for the males. So this is a female here. The males have a very distinct spot, hence the spotted wing Drosophila on the tip of each of the wings. And they're really, really obvious. If you were to look in the contents of a trap, you would see those spots actually pick out quite easily. I don't want to spoiler alert, but they are obviously on these traps too. The challenge, this was a graph that was provided by Erica's through Erica's work, that is trapping spotted wing over the course of 11 weeks, starting in July 1st. And what you see is the orange bars represent females within the trap, and the yellow bars represent the males. So what you see is, in all likelihood, if you're using males as your sort of criteria, you're looking at for detection of those males to make a decision, you should already be assuming that females are in that cropping system in that space. And we do know from past research that females will often move into the cropping systems kind of ahead of the males, especially in things like raspberries. So again, credit to Erica and Kat Benedict. So this year they did the work, collected all the data, and of interest, this was from their sampling in wild blueberry. Was interesting was that we didn't really see a substantial difference between sort of trap captures between the sort of solo cup and the sticky trap. So you know, I will say I did reach out to colleagues in BC, and there's a lot of published work out of Cornell Extension, so work that's been done in some of the counties in New York State, where they're sort of finding the same thing, where the efficacy of the traps is about the same as the solo cup, or the cup. Others use deli cups, but the cup and the apple cider vinegar. What I do want to draw your attention to is this is a little something that that I was curious to see if this was even possible. Is there any advantage to these traps at all? And one might be that you could actually take a picture of the trap with your phone, and then use kind of just, you know, magnify your screen. And it is obvious, and this was an older trap, but it is, the black spots on the tip of the wings is actually obvious. So this is just, according to Deb Moreau, kind of a comparison of the two traps for those that are kind of looking for more effective ways. From a cost perspective, the solo cups or the deli cups and apple cider vinegar definitely remain the cheaper option. For one thing, you can reuse the cups. The red sticky traps, certainly with the trace lures that we used, again, there are other lures that are available that come with jar traps. Definitely more expensive. The traps that were shown in the previous slide run about $60 for 10 lures and $16 for 10 traps plus your shipping. Ease of setup, both traps need stakes, you know, something to kind of suspend them within the crop. With the red sticky traps, you're dealing with tanglefoot, or the sticky part, which means you would need, you know, to be wearing gloves when you're putting these things on the stakes. These particular traps were actually quite large, and they can be difficult to work with. The lures are also really smelly, so you know that would also maybe have less appeal to some. Again, with the cups and the apple cider vinegar, you know, there is a small investment in time where you have to modify the cups to put the contrast tape in the holes, handles, everything like that. From a routine maintenance, there is a little bit of time savings, I think, with the red sticky traps, but marginal in that you still have to replace. You still really should replace those traps weekly. Especially if you're using those as a way to make a decision on sort of timing a spray, but also these are big traps, and you know, a word of caution is that in talking to colleagues, you know, they can catch other things that are kind of moving through those sort of, you know, low air currents. So yeah, so you would have to replace the trap weekly. The good news is the lures are good for a month, so they could stay intact. But again, with the cups and the apple cider vinegar, you'd be looking at a weekly change of you'd have to dump the contents and then recharge with more vinegar. So from a processing, if you're a grower and you just want to be able to go in the field and say yay or nay, I have them. So the nice thing with the sticky traps is you could use your smartphone. Whereas in the solo cups, you do need to dump them, unless you have way better eyes than I, but it could be something you could do quite quickly in the field if you just bought, you know, a kind of a shallow white bowl and dumped in, and again, in both cases, you can use, you know, handheld magnifiers, like just lenses. I didn't feel, based on the preliminary data, that there was any real difference. And in talking to colleagues in BC, they felt the same way. In fact, they actually cited that they saw improved trap captures with using the cups and cider vinegar early in the season. Work out of Cornell, certainly in the counties in New York State, also cited, you know, that efficacy was basically very similar. So from my standpoint only, this is again just my personal opinion. I think if there's possible advantages to one over the other, it would be that the sticky traps would give you, you know, potentially you could sample in remote sites. If you just had someone that could just get to access to the traps and image them, then you could make decisions remotely. And with the apple cider vinegar and cups, certainly the data coming out of BC and this was in high bush and raspberries, was that they had improved efficacy early in the season. So this is just a kind of a shameless plug for some of the work that is ongoing at AFC. So another trap that is being looked at is, we kind of, you know, endearing, we call it the sticky pie trap. So this is work that that is being developed out of BC, and it's been published, where they're using kind of machine learning. So the plan is that at some point a grower should be able to go up to these sticky traps, image them with the phone, and then then you would get feedback that would tell you what is the composition of the species or the things that are on your sticky trap. So what that meant in BC is they had to basically do a lot of collecting with sticky traps, and then people had to go through and basically take everything off those sticky traps and figure out what it was. And then they're working with the sort of cameras and the IA piece to figure out, you know, to reconcile what was actually on the trap versus kind of the image of the trap. So they're developing that technology. This was something that was developed probably 5 or 6 years ago for another invasive species, strawberry blossom weevil. So they're now using these traps as part of the organic cluster, looking at habitat amendments for multiple co-benefits in agri-ecosystems. So for me, I was interested in participating in this to put these out in remote sites, or sites just to see what we are collecting. So we're using a QR code, we're downloading it into Survey 123. And basically what we're doing is we're collecting all that data and working with colleagues. They're going to figure out the sort of composition of species that are in the east, so that hopefully one day that as this technology gets developed, we will be able to enjoy it here too. So then these traps could go basically anywhere. Right now we're monitoring in kind of hedgerows, wild bramble, but also commercial raspberries and commercial wild blueberry. So, and then the last, this is a deviation from spotted wing, but there are also a number of pests that we are working on here at AFC in Kentville. Most of them are invasives, so just kind of following up on spotted wing, but also Japanese beetle is on our radar, spotted, or strawberry blossom weevil. But something new to us is spotted lanternfly, and this is basically just a public service announcement. We were fortunate to get approval on a national AFC project that is going to be monitoring for spotted lanternfly in Canada. So there are people right across the country on this. This is a new pest. This is a fairly significant problem in the States. To date, we do not have established populations in Canada. It has certainly been detected in Canada, and there's been sightings and detection, and certainly in Nova Scotia, we've had 2 sightings, one in Halifax and one in Cape Breton in Sydney. A sighting is basically when we sort of did a search, we found somebody had uploaded images of dead specimens on iNaturalist, so more citizen science. And so we're kind of monitoring at that level. This is really a plug. If you see this past, see anything that looks like it, you know, continue kind of do the citizen science thing, continue to image, upload, report, please. This pest has multiple hosts. Its primary host in Asia is the tree of heaven, which is not really a big thing for us here in Nova Scotia. It's a bigger problem in BC. But it is known to use pine and red maple and other trees as a host. It's a hemiptera, so a true bug. So it basically is not known to attack the fruit of anything. And we haven't had any reports of damage to the trees that they're attacking, but we do know that it is anticipated to be a very significant pest in something like grapes. I don't know what, you know, in kind of those more, those smaller woody perennials. So I didn't find anything on high bush blueberry or raspberries, but the problem with this pest is that it is incredibly you know, its reproductive fitness is stunning. So it can pump out a lot of eggs. And when it feeds, it does other things. It's a honeydew producer, so it will make a mess of things. It will invite other pests like wasps and things like that will impact. So it's something that I just wanted to put on everybody's radar. There is lots of information out there at CFIA. They've got fact sheets, and certainly if you want to know more information, just reach out. So one of the things that we will be doing at Ag Canada out of Kentville is monitoring for it. And the image here is showing the traps that we'll be using to collect these. And hopefully in 2027, I will the person who's taking my position will announce that we have not had any. So with that, I would like to welcome Dr. Rachel Ripps. Rachel is taking over the small fruit berry entomology program in Kentville. So that would be wonderful. And massive thank you to Erica Bent and Kat Benedict for the work they did that I was able to exploit. Thank you.
Awesome. Thanks so much, Deb. Looking forward to meeting Rachel. That'll be good. So SWD, it's pretty well documented in small fruit. So you said blueberry, strawberry, raspberry, blackberry. Do we see it in grapes as well?
Not to the same extent. There's been lots of reports of its connection to vectoring sour rot. We trap it in grape all the time, and there's been some reports out of places like Italy and that where some thinner skin varieties may be susceptible.
Okay.
But any preference trials we've done, there certainly would take a raspberry over a grape.
Okay. And is there any evidence of them in things like tomatoes or cucumbers?
Not to my knowledge.
Okay. Hopefully just not, but and so just sort of to kind of connect between Caitlin's presentation, do we have similar sort of like flight time predictors for SWD and sort of those growing degree day?
So Oregon, so it's something I've been pitching for a very long time, is that we need to kind of develop more in-house models that fit our region. Oregon State University has a wonderful model. And so you can kind of plug our data in there. I've certainly been tracking annual heat units over since 2011 and kind of
Mhm.
first detection, first sustained captures. So to be honest, the Oregon model is actually reasonably tight.
Okay.
So there is data out there on that, that you could kind of anticipate when you could expect to see your first capture. I certainly know, when we first detected it, it was kind of late July, and it kind of bounced around there for a little while, but now we're definitely seeing it in June.
Okay. And I guess in the meantime, where we maybe don't have a Nova Scotia specific tracker, that could always be something we keep track of on our own farms. Through our scouting efforts, once we start to see that first, first few flies in our traps,
Yeah,
write the date down.
I would say early season monitoring is absolutely imperative.
Mhm.
It's a pest that once it's there, once it's in your crops, it's in your crops. It's going to bounce back and forth between all of the sort of non-crop, like the hedgerow and the wild host. But I think, early season, it's to get your, it's to be aware that now it's here.
Yeah.
And to start making, for growers to start making those decisions on best sprays.
Yeah, absolutely.
Monitoring is key.
And my final question, if you were growing in a smaller greenhouse, is netting an effective strategy to
Yeah, there's been
kind of
really good
exclude
data
them?
on using netting.
Okay.
But it is not, when I think of, some commercial farms,
Yes,
you
not
know,
quite feasible.
correct,
It's a
correct.
bit
It's not
big.
practical at all. And actually, there's
Yeah.
really good information on the cost effectiveness of mass trapping, because that's often a question I
Oh,
get.
awesome. Yeah.
Putting a lot of traps out and kind of overwhelming. Not that, it's there's a cost to that, and not just in time, right? And so when you look at that, say relative to the cost of malathion, for example, malathion is going to come out as the cheaper option.
Yeah.
If you start to kind of ratchet up to your more expensive products like Entrust, then the ratio changes. But again, the best thing would be for, growers to reach out. You can certainly reach out to myself or Rachel. But of course, Sunny's amazing in terms of, small fruit. And folks who do a lot of work with this, like Erica Bent, on, what's the best options for your crop.
Awesome. Yeah. So one last question. Is there a threshold number of flies before treatment for SWD?
It's all over the map. It really depends on the cropping season. You can go through the research. There's a number that say, if you see 10 males, to be honest, calculating economic thresholds for any insect species is basically a career for an entomologist. So that has not been, to my knowledge, that has not been done for SWD.
Okay.
So I think some, certainly there's work out of Maine that has done work specific to like wild blueberry, where they've said, they've done the calculation and said, yeah, X amount of males in your trap, spray. So I would really, I think I would reach, not to put Sunny on the spot, but if the grower that's looking for that, reach out to Sunny or reach out to me and just ask the question, what's in the literature for commercial raspberry, commercial, commercial blackberry. I would, one last thing that was kind of an interesting is the data on the red sticky trap. It did perform much better than the apple cider vinegar in tunnels.
Okay.
But it was very preliminary data, and I don't know why. So I think it leaves space to explore why that might have been a more effective. The catches were higher.
Yes.
It could have
I
been
think,
that the lure is just a more stronger attractant than the vinegar. It overwhelmed, but I don't know the mechanism.
yeah, I think it just goes to show that once we get into tunnel environments, we can't necessarily just take what works outside and put it in there.
Right.
We're going to see differences in varieties and traps. and all that good stuff. And I could
Right.
talk about it for ages. I really could. Um, but we are at 12:54. So thank you so much,
Apologies.
Deb. Nope, I could have shut you down, but I I was just so interested. So we're gonna switch over to Janelle. And yeah, kind of just keep those questions going. I hope everyone can maybe hang out a little bit longer. It's really good stuff. It's good stuff.
Yeah, I'll try not to make it too long here.
Sorry,
Okay.
Janelle.
No, it's totally fine. It's all good. All right, let's see if I do this correctly. Share. Slide.
Yeah,
Is
looks
it full
good.
screen for you there?
Yeah.
All right, perfect. Yeah, thank you so much for the invite. Really appreciate it. But again, my name is Janelle McKeel, and I am a PhD candidate at Dalhousie University's Faculty of Agriculture. And today I just wanted to provide a little bit of background information on what I'm doing in my PhD right now. And I'm focusing on, as my title says, the sustainable solutions for managing blueberry fruit fly. And so I'm still in the midst of my PhD, so I don't have any hard, concrete data to provide. But what I thought I would do is just provide some background information on the insect pests that I'm currently working with, and just trying to tease out those results that might make things a little bit easier for the blueberry growers moving forward. And so here we have the blueberry maggot, also known as a blueberry fruit fly. And so on the left, you can see that maggot alongside, right beside it, is the adult fly. And they're easily distinguishable from other insects that you might see in your field or on your traps. And that's because of that unique wing pattern that you see there, that W or that M shape. And the card on the right is showing what a blueberry maggot would look like inside your fruit. And in the middle of there, you see kind of that desiccated berry. And that's because the larvae or maggot was inside the berry, consuming the pulp and really drying it up. And that exit hole there is once the larvae was fully developed and exited the fruit. And on the far right-hand side, you will see what you would see with your naked eye if you were to find a blueberry maggot in one of the fruits that you're eating, but hopefully that is not the case for you. And so, as you might expect, blueberry fruit flies are found in blueberry fields. And wild blueberries specifically are Canada's most valuable fruit crop. And they actually account for almost 30% of all the fruit that's exported by the country. That said, blueberry production does face significant challenges from insect pests, and one of these is namely the blueberry fruit fly. And many markets, they have what's called a zero tolerance policy for the maggots to be in the fruit. So if maggots are found, entire shipments can be rejected by these export markets at the expense of the grower. And this is all to help prevent the spread into new areas. But Japan, the EU, and other major buyers, they have strict pesticide maximum residue limits as well. So products that growers used to rely on in the past, such as Fosmet, or also known as Imidan, those are getting phased out of use now. So all of these factors in combination are really, they make the managing of this pest all the more difficult for the growers. And here's just a simple life cycle depiction of the fruit fly. So when these flies, they emerge in the summer, and in Nova Scotia, that happens in late June, early July. They emerge from the soil and they find, honeydew or nectar to sustain themselves for energy. And then they very quickly mate. And once the blueberries in the field start to ripen, that is when they're going to start depositing their eggs in the berries. So they lay their eggs, and these eggs will hatch within a week or so. And these hatched eggs will be your larvae or your maggots. And these will immediately start feeding on the pulp inside the berry, drying it up as we saw in that first image in the first slide there. And so once these maggots are fully developed, they will then exit the berry onto the soil, dig into the first few centimeters where they will overwinter as a pupae, and this cycle will repeat itself year after year. And what's interesting about the fruit fly is that one female can deposit up to 100 eggs in her short lifespan of just a few weeks, but she will actually only deposit 1 egg per berry. And what she does is she'll leave behind a pheromone, which is like a pheromone deterrent from other females that will lay their eggs in that fruit to prevent that from happening. And so I was actually in a field this past summer, and we had just set out fresh new sticky traps, and we had revisited that field 2 days later. And on those traps in that one field, we got 152 fruit flies. So left unchecked, their populations can get out of control pretty quickly. And so in Canada, not all the regions that grow wild blueberries actually have this insect pest. But unfortunately, of late, we are seeing them slowly spread. And one example of this is the region of Quebec, which produces, most of Quebec's wild blueberries, never used to have this pest. And so they didn't have to monitor or spray insecticides. However, in 2018, CFIA detected fruit fly here for the first time. And in the subsequent years, they had found that it has established itself, unfortunately. So these growers are now having to monitor for the fruit fly, spray for it, and now they are also facing restrictions on their exports as well. Therefore, it is crucial that we have a sustainable integrated pest management plan in place. And so the themes of my research to help with this start with the development of a degree day model to help predict the insect's emergence. And I'm following this up with looking at how ecological factors may be influencing their populations, as well as testing reduced risk insecticides and investigating potential biological control options. So one of the big things that I'm working on developing, and is the main point of my talk today, is a degree day model for the blueberry fruit fly. And so many of you are likely already familiar with this, but basically these insects, they're exothermic, so they can't regulate their temperature the same way that you and I can. And so they depend on external heat to be able to grow and develop. And so every insect has a baseline temperature that they start developing at, the minimum temperature needed for development. And for blueberry fruit flies, this is around 5 degrees Celsius. So if the temperature is below this, the insect doesn't develop. It's just like hitting a pause button. And so each day we're going to add up the daily temperatures above this baseline to help predict the key life stages, such as when these insects are going to emerge. And using this allows us to be able to know exactly when we should be setting traps or applying our controls instead of having to guess. And so here is an example from a paper that discusses the emergence of apple maggots, which is in the same genus as blueberry fruit fly. And so what we're looking at here is that 50% of the population has emerged. And so we see in 1977 and 78, those dates are pretty close together, just a few days apart with their emergence. But then we take a look at 1980 and we see that it's more than a 2-week difference. And so there's a lot of variance that can happen between years. And a lot of this is down to the temperatures that we are experiencing on a year-to-year basis. And so if we did just fixate on those calendar dates, let's say if we were to say every year these insects are emerging on July 1st, we could get it wrong, because if we have a warm spring, they might emerge a little bit earlier, or a cold and wet spring, you might see that happen a little bit later, as this case here with the apple maggot. So by having access to these degree day models, the growers are able to get a real-time weather-based prediction that is rooted in data that is coming directly from their field. So it's so useful. And so in my research, I am developing this model for Nova Scotia. And to do this, I'm using the province's suite of weather stations, which many of you are likely familiar with. But these weather stations, they track things such as soil temperature, air temperature, wind speed, precipitation, humidity, etc., every 15 minutes of every day. And so what I've done is I've placed these monitoring traps along the edges in blueberry fields, and I record blueberry fruit fly captures 3 times per week from June until August. And I'm using 10 to 12 of these fields. And so from these weather stations, this is just my data from last year. I have graphed the average cumulative degree days from this past summer. But what we're interested in here is where does that fruit fly population fit in with these degree days that have accumulated? And so my model isn't fully developed yet. We're still collecting that data, so don't take this data as fact just yet. But using the data that I do currently have, this is what we're looking at here. So we're looking at the flies emerging around 520 degree days, peaking around 780, and dying off around 850. And so this peak that we're seeing here around that July 15th date, that's pretty significant because this is exactly when the blueberries are most susceptible to attack because they are ripening, and that date isn't quite when growers are going out and harvesting their fields, leaving a larger window for those fruit flies to attack and lay their eggs in the fruit. And so I'm going to be collecting this same data again next year to further validate and strengthen the model. But I'm very excited to be able to have this information for the growers moving forward. And so why is degree day modeling helpful? Just to recap, having these localized degree day models are going to help predict different points in the insect's life history. Like I said, when these fruit flies are going to emerge, when they're laying eggs, etc. It's also going to increase the efficiency and the precision of scouting efforts. And that's important because we're now seeing a shift from growers not managing just one field, but many fields in different areas. So it makes it very difficult or even impossible to be able to monitor all of these locations regularly. This can also help optimize management decisions. Like I said, when are we to apply insecticides on those key dates and not have to rely on the calendar dates. And in the end, we hope to have an app or a web-based application through Perennia, Perennia's website, to be able to notify the growers, okay, based on the weather station in your field, you're approaching this many degree days, which means that fruit flies are likely to emerge soon. So make sure you have your traps out and prepare your for insecticide application, basically. And so the degree day model validation was my main focus today, but I also wanted to just briefly touch on a few of the other projects that I'm working on. And one of the things that we're really interested in is understanding the influence of habitat features. And so we know from other studies that have been published with other insects that habitat is extremely important for their establishment and determining whether or not they'll even be in a field. Because especially with the fruit fly, when I'm sampling fields, there are some fields that have so many fruit flies that are infested, and then a field a few kilometers down the road where I get no counts at all. So there's something really interesting happening here. But things like field size, slope, alternative hosts, surrounding forest type can also have an effect. That is something that we're looking into. And so these two pictures here, both of them were actually taken in commercial wild blueberry fields, believe it or not. But the photo on the left, oh, sorry about that. The photo on the left kind of shows you, that's the ideal condition for blueberry growers, as the photo on the right, this obviously impacts yield and can reduce harvester efficiency as well. But what we're interested in is, do these weedy areas provide a refuge for these insect pests? So like I said at the beginning of the presentation, when the adult flies emerge, they're looking for nectar and honeydew to feed on. So are these flowering weeds, providing that for them, helping to boost their population even further? And these weedy patches with goldenrod or hair fescue, those taller weeds, they could also provide, a shelter for these insects, giving them a refuge against, wind, or making a more humid environment for them, compared to those open blueberry barrens that we're seeing in this picture on the left here, which could also help them, be sustained in the field for a longer amount of time, which wouldn't be beneficial to the grower, obviously. And so this is just some data that we're looking to continually collect this summer. And so I'm looking forward to analyzing that data as well. And over the past 3 years, I've also been conducting in-field insecticide trials. And this is to try to evaluate, different treatment options for the blueberry fruit fly. I'm testing already registered products, and this is to be able to confirm and compare their efficacy against each other, as well as testing new formulations that are in the pipeline with different insecticide companies, and even looking at a few different bioinsecticides, which are really interesting as well. And so to determine how effective the treatments are, I go and spray, and then come harvest from each plot, I'll go and take a sample of the berries. I bring them back to the lab, where I then gently crush the berries just to break open the skin. And then I soak them in a brown sugar solution. And this allows those maggots to float to the surface to be counted. Really feels like a elementary school science fair project, but it's pretty fun doing it. Anyways, these maggots, they're collected and then viewed under a microscope to ensure that they are in fact blueberry maggots, and then they are recorded in my data. So I won't be diving into all of these results today, but I am looking forward to sharing this data at future meetings. And one last project I wanted to highlight involves exploring biological control options for the blueberry fruit fly. And so in my master's research, I also worked in blueberry fields, and we found that crickets were highly abundant in our fields when we set out pitfall traps throughout them. But we really didn't know much about their role in the ecosystem. There's not a whole lot of published information on these field crickets. Other studies did find that these field crickets will readily consume spotted wing Drosophila pupae as well as apple maggot pupae. So one of the big questions I had leading from my master's into my PhD was, well, do these crickets also eat blueberry maggots? And so I conducted laboratory experiments this past summer where I placed live larvae and pupae on the soil surface, and then I introduced a cricket to the arena. And I found that, yes, they will readily consume both the larvae and the pupae. And then from there, I wanted to find out, well, the pupae are actually underneath the soil. So will the crickets dig up and consume those? And so I also did an experiment with this, and where I allowed the larvae to naturally pupate into the soil in a petri dish. However, I did not find the crickets to be able to dig up and consume these pupae, unfortunately. But this is an experiment that I'm looking to expand upon this summer and potentially conduct some in-field studies. And so just to conclude here, the development of my degree day model is well underway. I just have another field season left, and I think that it's really going to be a very useful tool for the growers. And we're looking to further investigate the influence of different habitat features, namely, those weedy patches. Are they having an effect on the populations of the fruit fly? There does appear through their studies to be some good reduced risk products available that work very well against the flies, as well as some of those products that possess low risk to the beneficial insects. Because not only am I testing, do these insecticides work against the blueberry fruit fly, but also testing, do they also kill our beneficial insects, such as our field crickets? So finding one that, targets the fruit fly, but doesn't target our beneficials. And lastly, continuing to explore those biological control options this field season as well. And so I know that was a rapid fire of some of the projects that I am working on. So if you have any questions, I'm happy to take them. And as well, my email is here. If you have any further questions, I'm happy to chat. And yeah, thank you so much for having me.
That was awesome. Thanks so much, Janelle. I appreciate the rapid fire. I think you did a great job. Would you be able to share that sugar solution so that people can try it themselves?
Yes, I can definitely. There's an article online that will give a detailed overview that I can send. Would it be best to send it to you, or?
If you actually want to post it in the chat, I don't know if you
Yeah, I'll
have
do
that
that right
function, whether you just share it to me or the group, and I can post it for the rest of the group.
definitely.
Okay, and just to clarify, you said you have one more season left for your degree day model. So is that going to be 2025 growing season as sort of
Yes.
your, okay, awesome. So maybe we'll see it for 2026.
Yes, yeah, I'm really looking forward to it. And it's
Awesome.
something that will probably have to be continually worked on, as you should with degree day
Absolutely.
models, but just having that baseline for a Nova Scotia model will be
Yep.
so helpful for the growers, I think.
Yeah, unfortunately, it's a lot of time, energy, and effort to kind of get those things going and keep them accurate. So it's definitely a
Mhm.
moving target, but it's great to have something we can start with. Absolutely.
Yeah.
Okay, so I appreciate everyone for hanging out, for the extra time. That's completely on me. I was just having so much fun that I forgot I needed to moderate. So there you go. A huge thank you to our 3 speakers here. We really appreciate you guys coming out, sharing your work. And I love seeing the themes across the different insects and crops that we were talking about today. I think that's always gets me excited, something that we can sort of all talk about and work together towards.